So good afternoon. Welcome again now this week. What I'd like to do with this afternoon session is to build a little bit on what I spoke with you about last Tuesday, where I was talking about the ligation. Actually, just before I start, can somebody please come online and just confirm for me that you're able to hear me and that you're also able to see my PowerPoint slide? Maybe can someone just come into chat and confirm you can hear me and you can see the slides? Brilliant, hearing and seeing. Thank you very much. So what I was talking about last week was this idea of getting some maybe genomic DNA and digesting with the restriction enzymes, and then digesting a plasmid with, you know, similar complementary cohesive ends, and then being able to clone or to ligate the DNA into the plasmid using DNA ligase. And in our examples, you know, I was talking about how we use PCR in order to create special restriction enzyme recognition sites that are most appropriate, you know, for trying to clone a fragment of DNA into a plasmid. And this is all very well and good in bacteria if we're trying to express a bacterial protein, so we can PCR amplify using the genomic DNA from the bacteria as a template for the PCR reaction. But this is no good for eukaryotic cells, because eukaryotic cells undergo post-translational modification. So they get a 5' cap, they get a 3' poly A tail, and they undergo this thing called RNA splicing, where introns that are located within the coding sequence of genes in the eukaryotic genomes, these regions get spliced out, leaving behind the exons, which are shown here in blue, that then get spliced together in order to make the mature messenger RNA. And it is the specific nucleotide sequence in this single-stranded messenger RNA that is of central importance in terms of producing an amino acid sequence in a specific protein that has a specific function. So it's not actually the direct nucleic acid sequence or the nucleotide sequence in the DNA that gives rise to the protein. It is the nucleotide sequence in the spliced mRNA that gives rise to an amino acid sequence in the protein. So when it comes to making a protein drug from a eukaryotic protein, we need to be aware of this splicing that goes on. Not just does splicing go on in order to produce mature messenger RNA, but there is this thing called alternative splicing, that depending on the cellular conditions, including, you know, a kind of nutritional situation or disease situation, you may even get alternative splicing of the RNA. So in the example that's shown here, on the left-hand side, we have our double-stranded DNA with 5 exons And during splicing, shown up here on the top, we get the removal of all the intron sequences, leaving behind the 5 exons that are then spliced together. This spliced messenger RNA then undergoes translation. And what we can see here is that the inclusion of all these different exons, including exon number 3, gives rise to a protein that includes all the associated functional domains, including this one here. In a separate set of cellular conditions, we may get alternative splicing where we exclude exon number 3, and in this case, the translated protein is not going to have that functional domain. So when it comes to expressing proteins, and from a research point of view, we're interested in the different functions associated with different proteins, but certainly from a protein biopharmaceutical point of view, we need to be sure that the protein that we're making as a drug for our patients includes all the required domains for functional activity in order to have its specific therapeutic protein. So not only do we need to be cognizant and aware of the splicing of RNA to give mature messenger RNA, but also we need to be aware of that whole issue of alternative splicing. So this is, I suppose, what we would see in terms of expression of eukaryotic proteins. And maybe here's the eukaryotic protein here that we want to make a protein drug from to give to patients. So we start with our genomic DNA. It contains intron sequences. These are then removed during RNA splicing in order to give the spliced messenger RNA, which would then be translated into giving us our protein sequence that we have here. So really, the nucleic acid sequence that we want to clone into a plasmid, to ligate into a plasmid, and then put the plasmid into a production organism that's going to make this protein. The sequence we're really interested in is this sequence here of the messenger RNA, not this sequence here that contains all the intronic nucleotides. So in principle, we would like to ligate and clone this single-stranded messenger RNA into the plasmid. The only problem is that the T4 ligase that catalyzes this reaction does not recognize single-stranded nucleic acid chains. It only recognizes double-stranded DNA chains. So in this case, what we have to do is convert this single-stranded messenger RNA into double-stranded DNA. We need to reverse transcribe it. And we can do this using an enzyme that's purified from retroviruses, an enzyme called reverse transcriptase. And this makes what's called complementary DNA, or cDNA. So how does this work? Well, instead of using the genomic DNA as the template for the PCR reaction, or using messenger RNA, which isn't a substrate for a PCR reaction, we now use our complementary DNA as the template for a PCR reaction. So over here, in terms of our PCR, the DNA sample, the template is cDNA. Again, we're going to use primers that contain the additional nucleotides, so that it's not just complementary to the target cDNA, but contains additional nucleotides that represent the specific restriction enzyme recognition sites for particular enzymes. So now we're going to get a PCR product that, in addition to the cDNA sequence, is going to contain additional restriction enzyme recognition sequences. This can then be digested by restriction enzymes, and now it has these nice complementary cohesive end hooks that I spoke about last week that allow for cloning into a plasmid. So here we have our PCR product. Here we have a digested PCR product. Here we have the digested plasmid. This can then be ligated by the DNA ligase to make plasmid. Initially, we would transform the plasmid into bacteria. We grow up lots and lots of bacteria, and we can purify lots and lots of plasmid, and we can then put the plasmid into our production organism, and the production organism is going to make the protein of interest. So what kind of proteins are we interested in expressing? What therapeutic proteins are we interested in expressing using this sort of technology? Well, my colleague, Dr. Gahan, spoke with you at a fair bit of length about insulin. So I'm not going to go through this in a huge amount of detail, but insulin, it's actually encoded by a single gene that is translated at the rough endoplasmic reticulum. So the protein that's produced has a signal sequence which is removed. Then in the reducing environment, in the oxidizing environment of the endoplasmic reticulum, we get the formation of disulfide bridges and proteolytic cleavage away of the C-peptide in order to generate a heterodimer. So insulin is a heterodimer. Now, I suppose the key thing about the production of insulin by biopharma companies is they found that you can put the nucleic acid sequence that codes for the A chain into one plasmid, put it into bacteria, and get the bacteria to make the A chain. And independently, we can put the nucleic acid sequence that codes for the B chain into a second plasmid, put that plasmid into bacteria, and get them to produce the B chain. Now, it would be exceptionally rare for us to be able to bring together two proteins that are going to make up a heterodimer and get them to form the correct disulfide bridges so that it creates a functioning enzyme or a functioning protein. But because of the fact that the A chain and the B chain are so short, it is possible to purify these two chains and to chemically create these disulfide bridges and produce active recombinant insulin. So I suppose this is not necessarily a great example of proteins that can be expressed, because this wouldn't normally happen. This wouldn't work for most proteins if you were trying to bring together two different subunits. But certainly for insulin, this actually does work. The other thing I think that's noteworthy about insulin is that once we have the coding sequence, and this applies not just to insulin, it applies to any nucleic acid sequence encoding any protein that is cloned into a plasmid, is that we can actually mutate the coding sequence so that when the nucleic acid sequence is transcribed and translated into protein, it creates a different amino acid at a specific location within the recombinant protein. So this can be used for a variety of different purposes. For example, it can be used to characterize protein function. Over here on the right-hand side is actually an enzyme that I was working on as a postdoc in the US. This is the enzyme histidine decarboxylase. It makes the modified amine histamine, which you'll all be familiar with from the point of view of pharmacological importance. Histamine is very important, and histamine receptor antagonists are very important. And what we were doing was using this three-dimensional structure. We mapped amino acids that we thought were being of particular importance for the catalytic mechanism. And using this sort of a site-directed mutagenesis approach, we altered the nucleic acid sequence in a plasmid, and then we transcribed and translated the plasmid into protein to see how that affected the enzymatic activity of the protein. So we were able to identify the amino acids that are critical for histamine biosynthesis. But there's lots of things that we can do with this sort of mutagenesis. We can investigate protein folding, protein characteristics. We can alter catalytic activity. We can alter protein stability and protein solubility. So I'm going to talk about that again now in a moment. I suppose this is of particular relevance and particular importance within the biopharma industry, because we would try and get proteins that would be soluble in aqueous solution. We don't want them forming aggregates and lumps. So very often the formation of these lumps, very often the formation of these aggregates, has to do with the fact that there are hydrophobic amino acid residues located on the surface of the protein. These don't want to be in solution, so they form aggregates. So we can mutate the nucleic acid sequence that codes for the amino acid sequence to produce hydrophilic amino acids on the surface of the protein. And this then improves the solubility of these therapeutic proteins. And a key example of this actually relates to the insulin that we've spoken about. And one of the most common forms of insulin that's now available is this insulin glargine. And there are a series of mutations that have been introduced into the nucleic acid sequence that codes for the proteins, the insulin A and B chains, in order to improve the therapeutic protein or the therapeutic insulin as a medicine. So in terms of treating diabetic patients, this insulin is even better than if we were to put in just the human insulin. So why? How does this happen? Well, changing amino acid residue asparagine at position 12 in the A chain prevents the deamidization of insulin, and therefore it slows down the degradation or the breakdown of the protein drug that's functional. The other thing then that it does is to through this sort of technology, they've added the amino acid arginine to the B chain, and this makes the molecule more soluble at a specific pH. So once the protein goes into a solution at that particular pH, it is clear, so it is injected into the patient as a clear solution. But then once it gets into the patient, there's a change in the pH to neutral pH that promotes the aggregation of the insulin. Okay, so it forms this sort of an aggregate here. And what this means is that there is a slow deaggregation with the release of individual insulin monomers, kind of one at a time from the aggregate, leading to a slow peakless release of the insulin. So insulin is always being, I suppose, active, rather than, previously our diabetic patients had to inject insulin after every meal. Now it's possible with this product to have one daily injection of the insulin. Okay, so insulin is an example of a type of recombinant protein drug that we might want to produce. It is produced in bacteria, and it says what we have is we can independently produce the A and the B chain and then bring them together. Other examples then of proteins that are produced or that have a therapeutic value, there's a table of them here. Without going through all of them, I suppose I would just make reference to this first one here, EPO. So I suppose we'd be kind of familiar with this in terms of doping and athletes, but in terms of its clinical use or its therapeutic use, it can be used to treat anemia and promote the formation of red blood cells that will carry oxygen. So this is another example of a recombinant protein. But I suppose somewhere along the line, we also began to appreciate that we could, in addition to the mutagenesis that I've spoken about now already, we can engineer other completely novel proteins that bring together quite discrete functions from two different proteins, very often from different species, bring those together into a single protein with a novel and unique therapeutic value. And we can do this by getting the cDNA sequences that code for these different functional domains from different proteins. You can see over here that we have cloned multiple nucleic acid segments into our plasmid, bringing together bits of genes from lots of different proteins from different species in order to make a new protein with a new function. Okay, so there are two applications on this: production of chimeric proteins containing discrete functional domains that have added therapeutic value, and also the production of protein chimeras, or engineered proteins that have tags that assist in the detection and purification of proteins. Excuse me. So let's I suppose, briefly speak about two examples here in relation to this. I'll talk about these two examples. The first is the production of a chimeric protein, or an engineered fusion protein, that mixes and matches functional domains from different polypeptides. So in this case here, I'm going to talk about DAB389, and this is used in the treatment of malignant cutaneous T-cell lymphoma. So these particular cancer cells express a component of the IL-2 receptor. So this is a receptor, which is shown in blue here, that's expressed at higher levels on the surface of these cancer cells. What this drug does is it's a fusion protein that brings together the residues, amino acid residues 2 to 133 of human IL-2. So it takes this part of the IL-2 protein and it fuses it with the first 389 residues of a toxin. So this is a natural toxin, the diphtheria toxin. It effectively brings together these two functional domains into a novel single protein. And it does this by cloning the cDNA that codes for IL-2 and the cDNA that codes for the diphtheria toxin into a plasmid, so that it now has a single nucleic acid sequence that codes for this novel engineered protein. So when we put the plasmid into the cells, instead of making two discrete proteins, it creates a single protein with two functions. The targeting domain is IL-2, which is orange here. So the orange bit here is IL-2. And the fusion protein also has this effector domain, which is the diphtheria toxin. So in this way, we can use this engineered protein in order to treat cancer. The second scenario where it is important or is beneficial and advantageous to produce an engineered protein or an engineered fusion protein is in the addition of tags. And the example that's shown here is where we would have cloned the nucleic acid sequence that codes for green fluorescing protein into a plasmid. And then we put the plasmid into bacteria, for example, and bacteria will express this green fluorescing protein, or there's a number of different red and blue and green variants on this. And while that would be a very kind of a simplistic application, what we can do is we can clone GFP in as a fusion protein, another protein that is a skin protein, and clone this into a plasmid that contains the promoter region to ensure that this particular fusion gene is expressed in skin cells. And what you can see here is transgenic animals, transgenic mice that were made from this type of an engineered protein strategy that allowed us to identify which pups, which transgenic mice are actually producing and expressing this fluorescing protein in their skin. And you can see here that this pup here isn't expressing that protein. So while it is most visually appreciated here in terms of our transgenic animals, this approach can be used in order to ensure the correct targeting of protein drugs within a patient. So we can make a fusion protein containing our recombinant therapeutic protein fused to green fluorescent protein. We can put this into the patients, and we can, or in a kind of a clinical model, a toxicology model where we're doing research to get the correct localization of the protein drug, and it will allow us to readily detect the cells and organs that the drug is being targeted towards. Down here at the very bottom, this is just an example from my own lab where I created a chimeric protein with this histidine decarboxylase enzyme that makes histamine. It's normally located to the endoplasmic reticulum, so I made a fused protein that is part the enzyme that makes histamine, part GFP, and you can see here quite clearly how the enzyme is localized to this reticular structure of the endoplasmic reticulum. Another application of this type of protein engineering is, and the addition of tags to recombinant protein, is used very frequently by the biopharma companies. So we have a company down the road here in Cork, BioMarin. It's using this in order to purify up recombinant proteins that are going to be given to patients. So it's based on what's called IMAC, or immobilized metal affinity chromatography. So chromatography means we have a chromatography column, and we have some sort of agarose beads or cephalose beads, carbohydrate beads, that are used as the stationary phase in order to pack the column. So these are our cephalose beads. Down here you can see them here. And conjugated to these cephalose beads, which are again the beads that are packing our column, there's this nickel NTA, or nickel nitrilotriacetic acid moiety, that's conjugated onto the beads, and this specifically recognizes and binds to an imidazole ring. Imidazole. And this imidazole ring is naturally present in the amino acid histidine. So as is shown here in this example, we have a protein here, and at the end of the protein we have 6 histidine amino acids, and through these histidine residues we get a very specific interaction between our recombinant protein and the affinity column over which we're trying to purify the protein. So how do we get our recombinant protein here with 6 histidine residues at the end of that protein? Well, we can do it by taking the cDNA for the protein that we're interested in, the protein that we're going to make into our therapeutic protein, and we can, to this cDNA, we can add the nucleic acid sequence that codes for 6 histidine residues. So this specific nucleic acid sequence is CAT, so there's 6 of them there. We put this in at the end of our cDNA and we ligate it into the plasmid. We take that plasmid, we put it into our production cell, and it produces this recombinant protein with a histidine tag. We can then take the protein produced by our maybe CHO cells, for example. We can load it onto our affinity chromatography column. The protein, our protein of interest, which has the histidine residues, now interacts very specifically with our chromatography column. So our protein of interest is retained on the column, all the other proteins go through, and then we can elute our therapeutic protein in a relatively pure state. We can elute that off of the column. So another scenario where we might like to exploit engineered proteins is in the area of monoclonal antibodies. So monoclonal antibodies are now being extensively produced in the treatment of disease, or for the treatment of disease. Again, we have a number of companies here in the Cork area. We have Johnson, Janssen, we have Lilly, we have Merck, we have Regeneron. We have lots of companies here that are making these monoclonal antibodies for use in patients. Now, as you'll know from your immunology, antibodies are made by B cells. So an individual B cell clone has within its genome the genes that code for a heavy polypeptide chain and a light polypeptide chain. So these genes get transcribed and translated into protein, and then two heavy chains and two light chains come together in order to make this Y-shaped antibody. So a single B cell clone produces a single antibody that recognizes a specific target. A different B cell clone has different nucleic acid sequence in the genes that code for the heavy chain and the light chain. So now this second B cell clone produces, when the genes get transcribed and translated, they produce an antibody with a slightly different ability to recognize and bind to an antigen. So in terms of the antibody, the bit that recognizes the antigen target is up here at the tips of the Ys in what's called the FAB region. So there's about 30 amino acids in the heavy chain and 30 amino acids in the light chain. These come together in order to form the antigen binding site for a particular antibody. So for B cell clone 1, for example, there will be just a few nucleotides at the end of the gene that codes for the heavy chain and a few nucleotides at the end of the gene that codes for the light chain that are going to be different depending on whether it's this B cell 1 or B cell 2. So it's those few nucleic acid change differences that account for the ability of antibodies produced by different B cells to recognize different antigen targets. Now, when it comes to the pharmaceutical industry or the biopharmaceutical industry, they're interested in producing monoclonal antibodies. So these would be ones that are all derived from a single B cell clone or a single hybridoma clone. Again, I think you've probably done this as part of some of your courses that produces the production of hybridoma cells. And in this way, the biopharmaceutical companies are capable of producing just a single antibody that can be purified and then used as a drug. So rather than working with B cells that are derived initially, these studies are these drugs are developed in mouse cells. Rather than using mouse cells and mouse B cells to produce mouse monoclonal antibodies, or even rather than using mouse hybridoma cells to make monoclonal antibodies, what the companies now do is they take the cDNA that codes for a heavy chain of an antibody and the cDNA that codes for a light chain of an antibody. They clone those into plasmids, and the plasmids are put into, normally it's a CHO cell. The CHO cell transcribes and translates those plasmids into the heavy and the light chain of the antibody. The antibody, the heavy and light chains then come together in the form that Y-shaped structure. And these are secreted out into the medium, and the companies then collect the medium that the cells are growing in. But they don't use B cells, they don't use hybridoma cells, they use this kind of recombinant DNA and recombinant protein, engineered protein technology in order to achieve this. Now, when it comes to recognizing antigen, I said that the bits that are important in the antibody are right up the very tips of the Ys. And this involves about 30 amino acids from the light chain and 30 amino acids from the heavy chain coming together in order to create this three-dimensional region that's capable of recognizing an antigen. So this is like our FAB. In principle, for a lot of what we want to use these drugs for in a therapeutic context, we actually don't need all this bit down here. We don't need the rest of the antibody. We really only want the bits that are capable of recognizing an antigen target. So that involves 30 amino acids from the 30 amino acids from the light chain and about 30 amino acids from the heavy chain coming together now to form that FAB region that recognizes a specific antigen target. So now, very often, depending on what drug the companies are trying to make, rather than taking two plasmids, and very often, in fairness now, you can incorporate these into a single plasmid with what's called an IRIS sequence. But what the companies are doing is they're taking the cDNA that codes for this kind of a single fragment, so that when it's transcribed and translated, part of the translated protein has amino acids from the light chain, part has amino acids from the heavy chain, and that this is actually sufficient in order to recognize and to bind to a particular antigen target. And there are some instances where this is preferable compared to production of the entire monoclonal antibody. So on this slide here, we have some of on this slide here, we have some of the applications associated with these sorts of engineered antibodies. So here's our full monoclonal antibody here, our mAb. But engineered, I suppose, applications of antibodies and this SCFV. So SCFV is the name given to these shortened fragments, signal chain fragment variable, SCFVs. What we can do is we can, I suppose, clone the nucleic acid sequence that codes for cytokines. So these are proteins of the immune system. We can clone these in frame with the heavy chain of the monoclonal antibody. So now, instead of just having an antibody structure, we now make proteins that have, as part of chimeric proteins, they have cytokines in frame here. So what this does is has an immune signaling molecule attached to an antibody that recognizes and detects a target cell. And again, in this case, what we're doing is cloning the nucleic acid sequence in order to engineer a completely new fusion protein that's part antibody, part cytokine. And what this does, is particularly for cancer cells, is it improves immune signaling within the locality of the tumor. An immunotoxin here, we could see an application of we spoke about previously, where we get the diphtheria toxin, the nucleic acid sequence that codes for the diphtheria toxin, and we clone this, we ligate it in frame downstream from the heavy chain of a monoclonal antibody. So now we produce a fusion protein that has both a heavy chain and the diphtheria toxin associated with it. Other engineered variants of antibodies shown over here on the very right hand side, we have bispecific antibodies, which are actually composed of two different antibodies. So here we have a heavy chain and a light chain from one antibody, and a heavy chain and a light chain from a different antibody. And we can actually get these to come together to make a new antibody that recognizes two independent targets. And in this case here, what it does is it brings together an immune cell with a cancer cell. So there's lots of different applications here. Look, and some of them, there's a bit more detail given here in this slide in relation to what each one of them actually involves. All right, so up to now, as part of this discussion, I've spoken about therapeutic proteins, things like insulin, EPO. I've talked about monoclonal antibodies, and I've talked about SCFVs. Other types of proteins that we might want to express or to engineer include some that are currently being using as vaccine preparations. So this slide here, I suppose, summarizes the different types of vaccines that are currently being used, such as live attenuated pathogen, inactivated pathogen, are where we grow up a microbe and then purify a specific subunit from that microbe that can then be used to vaccinate human patients. And I guess the classical example here now at the moment is we could grow up our COVID-19 and we could specifically purify the spike protein from COVID-19 and then inject this spike protein, and patients would develop immunity based on this. But the other thing that we can do is we can take the nucleic acid sequence that codes for the spike protein, we can clone it into the plasmid, so we now have recombinant DNA. We can get a cell type to express this recombinant DNA to make a recombinant protein of the spike protein, and then inject that into patients in order to develop immunity. I suppose the final type of vaccine that's shown here on this slide that I'm not going to talk about here, but it involves potentially using DNA and RNA viruses. So that would again be very topical at the moment in terms of our response to SARS-CoV-2. The final type of engineered proteins that I want to talk about here now are called chimeric antigen receptors. And these are based on the activity of CD8 positive cytotoxic T cells, which normally function through a T cell receptor to recognize and bind to microbial antigen, shown as the triangle here, being presented by MHC class I molecule. So when this happens, when the T cell receptor recognizes this microbial antigen, very often from a virally infected cell or from a cancer cell that is expressing a tumor specific antigen, what happens here is that we get degranulation of the cytotoxic T cell, which then acts to release these perforins in red, which bore holes in the surface of the target cell, and these little blue granzymes that go in and destroy the target cell. So this is how T cell receptors normally work. They also function in the context, as I said, of cancer cells that are expressing tumor-specific antigens on their surface. But very often our cells don't process the tumor-specific antigens and express them in association with MHC class I. So this new, I suppose, approach is to make a chimeric receptor that is part receptor and is also part of an scFv. So here we have a single protein that is capable of recognizing antigen and activating the T cell. So the way this works in practice is that we get T cells from patients with cancer. We transfect in plasmids that contain these chimeric antigen receptors. So this is a plasmid into which we have cloned the nucleic acid sequence that codes for the scFv, are the components of the T cell receptor. We put this in, the T cell expresses this new chimeric receptor. We grow up millions and millions of these T cells. They get infused back into the patient. And in this case, the CAR T cells recognize, bind to the cancer cells and destroy the cancer cells. So that's my last slide in terms of the presentation now today. Look, I think we've gone right to the very end in terms of time. So it's bang on 2 o'clock my time, so it's bang on 3 o'clock your time. I don't think we have time for any questions, but I suppose just as last week, if anybody has questions in relation to some of this content, has questions about some of these genetically engineered proteins that I've been speaking about, please feel free to maybe write your questions to Professor Marwa, and she can provide you with my email address, and I'm more than happy to take questions. Okay. This is the last lecture session that I will do with you. So best of luck with your studies, and good luck with your biotechnology module. Okay. Thank you very much.